request everyone to kindly switch off your phones, please, because it gets disturbing for the speaker or the performer. Good evening all. On behalf of Sarojini Hegde and the entire Hegde family, I welcome you all to the 8th edition of the Professor KTM Hegde Memorial Lecture. To those of you who have not known Professor Hegde, he was a professional who dedicated the 40+ years of his academic life to the study of archaeology and ancient history of the Indian subcontinent. An MA from the University of Cambridge and a doctorate from MSU Baroda, he also had multiple fellowships for teaching and research at several respected academic institutions, including St. John's College, University of Cambridge, England, Harvard University in the US, and Wolfson College, Oxford University. He has published over 70 research papers in well-known Indian and foreign research journals. He is the author and co-author of a number of books on ancient metals, prominent among them being An Introduction to Ancient Indian Metallurgy and Early Indian Metallurgy. Under his leadership as Head of Archaeology and Ancient History, and later as the Dean of the Faculty of Arts at MS University, the Archaeology Department enjoyed global academic interest and support. The Ford Foundation in New York recognized the work and offered a grant which boosted the scientific tempo and progress of the Department of Archaeology. It is in dedication to this pursuit of academic excellence and his deep appreciation of the arts which prompts us to invite our guests who help in the celebration of the human spirit. Today, we are privileged to have our eminent guests who have kindly accepted our invitation for this memorial. Our first guest is Dr. Satyajit, who is a renowned cell biologist who will acquaint us with the intricacies of the human cell and more about some of the scientific organizations that he works for. Our second guest is the well-known vocalist Padma Shri Shubha Mudgal, who will uplift us today with her music of the mystics. Ladies and gentlemen, request you to welcome our guests with a round of applause. A brief introduction of Dr. Satyajit. Dr. Satyajit has his roots in our very own Vadodara. He is currently the Leverhulme International Professor at the Center for Mechanochemical Cell Biology, University of Warwick, and Distinguished Professor at NCBS, Tata Institute for Fundamental Research, India. From 2013 to 2023, he served as the Director of the National Center for Biological Sciences, a center of the Tata Institute of Fundamental Research in Bangalore, India. He's also the founder of the Bangalore Life Sciences Cluster and served as its chairperson. He was the director of Institute of Stem Cell Science and Regenerative Medicine and Autonomous Institute of the Department of Biotechnology, Government of India. Dr. Meher studied chemistry at the Indian Institute of Technology, Mumbai, India, and obtained his PhD in life sciences from the Rockefeller University in New York, US. In his laboratory at NCBS, he studies the fine structure, function, and dynamics of the membrane of living cells using methods derived from biology, chemistry, engineering, and physics. His work has provided a new understanding of how membrane structure and composition are regulated in living cells, and a deeper perspective of the functioning of the roles of non-clathrin endocytic pathways. I'm sure he'll demystify some of these for us. He is an elected member to the following prestigious national and international societies: Fellow of the US National Academy of Science, Fellow of European Molecular Biology Organization, Fellow of the World Academy of Science, Trieste, Italy, Fellow of Indian National Science Academy. He's also the recipient of several major awards. In 2016, the National Order of Merit of France, 2013, Distinguished Alumni of IIT Mumbai, and several other prizes. We are very happy to welcome Dr. Mayer.
Well, first of all, let me, honestly thank the Hegde family for inviting me to come and speak today. I don't often give public talks, and I'm not sure what Harsha had in mind when he wanted me to come and speak to you this evening. If while we get the projection up, I thought it would be really fitting for me to talk about some of the institutions of science that we have been involved in, but I'll also give you a glimpse and a sense of some of the work that I've been doing. It's also very in some ways very poignant to come back to Baroda and speak here because I grew up in Baroda, not so far away from here, and it's been almost close to 45 years since I have actually given a talk in Baroda in a public space. Of course, I've been to the university and spoken in the university, but I haven't really spoken publicly. And when you grow up in a place, you always wonder who you're going to embarrass. Even the projector went to sleep.
Awaken it soon.
But to be quite honest, I'm actually terrified because I'm between you and Shubha Murga, who I've also come to listen to. Right, here we go. So, the title of my talk, I gave as a way to find out who is really interested in understanding something about the cell. And I can see there's a large number of people here, and I'm incredibly grateful for having such a big audience. Oh, and close friends as well, makes it even more difficult. But let me start. So, given that I'm giving a lecture in the KTM Hegde Memorial Lecture Series, about the archaeology of, and who was a renowned archaeologist and an incredible academician, whose work, I think, as we just heard, has been all about academic excellence as well as an engagement with the arts. I thought I would try and do a parallel number. So let me just tell you what I'm interested in. I mean, here is something that many of you may know or may have seen. It's a neutrophil which is floating about in our blood, navigating the red blood cells and hunting for bacteria. So this is why we are not being continuously consumed by the bacteria that's sitting around everywhere, and we get infected, we don't get septicemia. It's because of these creatures in our blood, neutrophils, that go and do this. The amazing thing about the neutrophil is that, well, on any cell, is that what you have just seen is the cell, almost being able to follow this bacteria. And if and this is in real time. It's a movie that was also collected a few years ago, in fact, even before I was born. But when the cell is following this bacterium, it ends up, steadfast in its purpose and goes and grabs it and swallows it. This entire process of getting hold of this bacterium, it obviously has to sense it, meaning it has to smell it, or sense chemicals that have been given by this bacterium. It needs to know it shouldn't swallow the red blood cells because that would be harmful for the system, but it should only go after the bacterium. And the question is, how does the cell make all these decisions? And the decision that the cell makes is being made at the level of the cell membrane, which is the outer surface that covers the cell. It's a two-dimensional fluid sheet of lipids in which proteins or sensors are embedded. And this is fundamentally is an information receiving and processing hub. It's something that senses and responds and gets the cell to respond in real time. In fact, if you cut the cell and get rid of most of it, get rid of its nucleus, get rid of, maybe not its mitochondria, but get rid of the nucleus, this little cytoplasm that you build, it will do exactly the same thing. It will go around and capture these bacteria and make a decision about, saving the rest of the organism. So the question that we have been asking in my lab is, how does the cell do this? And I'm not going to give you an answer right now, but I'm going to tell you, this is the motivation in some sense for trying to build an understanding of the surface of the cell. So the title of my talk, The Journey to the Surface of the Cell, in some ways is evocative of our trying to understand the surface of the cell and the journey to understand that surface. But before I get there, I want to tell you something about where I'm doing this research and the institutions and the scientific organizations that have allowed me to pursue this kind of, I would argue, if some people would argue, esoteric, pleasures of engaging in scientific pursuit. So the story, what I'm going to tell you about is the story of the National Center for Biological Science, and how it's a sort of a dream that came true, and how this also led to the building of India's first life science cluster. And for that, I think it's very important we understand some bit of history and context, and why this is also important to us as scientists and citizens, especially now. I could just draw a timeline and say, well, there was Professor Siddiqui. In 1984, he said, he went to the Planning Commission and said, we need a Center for Biological Science. In 1991, it was formally set up as the director, with Obaid as the director. And then he got Vijay Raghavan to come and join this center, and he made him the director, and then he recruited, got me kicking and screaming to come and head this place. So I could end here and say, well, this is how we set it up. But I think I want to go a little further back and tell you what happened. So in fact, the story of NCBS begins with Baba, Homi Jahangir Baba, who establishes the Tata Institute for Fundamental Research as a way of building what I would call different kind of temples then. These were temples of modernity that were being constructed by, even before independence, when it was seen that we needed to build a scientific engagement and a scientific temple in this country. And Homi Baba built the Tata Institute for Fundamental Research. I started at the old yacht club, this beautiful building, which now is still part of the Department of Atomic Energy. And the Tata Institute of Fundamental Research gave rise to the Department of Atomic Energy, in fact. And in 1962, this was the TIFR. It was a fabulous building built by a French architect at the seafront. In fact, the sea is right at the back there. And I mean, this building is really an extraordinary architectural feat. And this was a picture in 1962 when the entire TIFR had started out there. But 1962 was also when the era of molecular biology, which was distinctive from the previous eras of biology, or where it was more observational and descriptive, the idea that one could understand the cell and could understand molecular mechanisms inside every single living cell that we encounter, including ourselves. This had just begun. The structure of DNA had just been made available. Nobel Prizes were being given for this for Watson and Crick, and unfortunately not to Rosalind Franklin, but the structure of DNA was understood. And biology was being also reduced in some ways to an understanding of chemistry. So it was in this backdrop that we discovered this letter that was written by Homi Bhabha to Dr. Siddiqui, where he says, why don't you come and build the new biology in a place for physics, mathematics, chemistry. I mean, this is an extremely interesting situation where you're bringing somebody who's, I mean, by then, Obaid had already created some ripples where he was. He was at Yale, and before that he was in Glasgow. And a very famous physicist had told Bhabha that if you want to bring somebody to build modern biology, you should bring Obaid. So Obaid was invited by Bhabha, and he decided he wanted to come back. And I guess there was a new era being born in India, and he wanted to come back and contribute. So Obaid arrived in 1962 in India and began to build the molecular biology unit in TIFR, Bombay. That's Obaid. And this was when he realized that the kind of work that he was doing would need to undergo a sea change because the context in India was distinctive. And so he began to build appropriate systems by which one could begin to understand the new biology from its molecular perspectives in TIFR, in Bombay. Also in a situation where he's working within this physics institute that is spawning atomic energy, the atomic energy program. So he decides that he will take on behavioral genetics and try and look for the molecular basis for behavioral genetics. And this was, I think, a very prescient move, because very few people in the world were doing this, and he said he could combine genetics and molecular biology and begin to build this program in Bombay. Now this was very exciting, but this didn't satisfy Obaid fully, because he dreamed that he would build an institute that would look at biology across all its scales. So if one wants to understand biology, I mean, that is a living, breathing system, a living organism, you really need to build it up from its molecular constituents to the constituents that make it, breathe and talk and walk and speak. So he decided that he would look at building this institute, an institute which would have people that worked in biology across all scales, from molecules, that is at the scale of physics and chemistry, to genetics and evolution. And this was a diagram from 1993. In fact, at a faculty meeting, it turns out, one of the few first faculty meetings that he had, where he builds these different divisions. There's molecular and cellular biology, he says. There's developmental biology and genetics, there's neurobiology, and other areas. And here he brings in something called theory as well. So he's obviously trying to connect biology and bring in a modern understanding into the system here. And it starts in this very wonderful little space which had 4 rooms that were dedicated to the life sciences at the Tata, what was called the Tata Institute in Bangalore, which is actually the Indian Institute of Science in Bangalore. the National Center for Biological Science of the TIFR begins in this space. You have, at this time in 1992, we had, the NCBS had already started, and Vijay had been recruited to become, I believe, the next director at NCBS at that time. And a bunch of really exciting young colleagues also joined in, on this adventure that Obaid, in some sense, to go to the promised land of biology across scales. And in fact, this was a unique experiment. There were not so many places anywhere in the world that were thinking about looking at how do you study biology in this way, in these different scales. And I think this was what Obaid went around selling to people he wanted to recruit, to these 4 little rooms, which had almost nothing in it, at one corner of the Indian Institute of Science, which also didn't want to do very much with this intervention from the outside. With the Indian Institute of Science, huge campus, amazing institution, didn't want this carbuncle dumped on it. But anyway, it was there, and Obaid, started recruiting people. In fact, this is when he contacted me sometime in 1993, and Obaid met me in a restaurant in New York, where I was doing a postdoc at Columbia University, and he says, "Look, whatever you do here in this country, he was there, of course, 30 years before, will be a drop in the ocean. But if you come back and work in India, it will make a huge contribution. And we have this fantastic institute that we have made." Of course, he didn't tell me there were 4 rooms with nothing in it. And amazing people you can work with, and anything you want will be made available. I said, well, let me come in. Obaid was very persuasive, and I said, well, this sounds very exciting. And at that time, I must say, I had already been in America for 10 years, and I wasn't really considering coming back to work in this country. So, but there were things that were happening at that time. There were these winds of terrible changes that I was listening to, and I felt that I needed to come back and understand what was happening in our country, unless it became completely alien to me. So I came back to live and work in Bangalore in these 4 rooms, and I must say, haven't looked back since. I mean, it has been an incredible journey, and I'm going to take you through that journey now. I've had some fantastic colleagues over the years, and the atmosphere and the culture of engagement that Obaid had generated in this place of collaborative working spirit was just extraordinary. So all of us were working at different scales of biology. So in principle, we should not have understood each other. I mean, you can have, you have departments dedicated to molecules or genetics or development or biochemistry, or and people don't talk between departments. But here you have people working in all these different areas of biology, just talking to each other all the time, and trying to understand each other's language, which was really very exciting when you come and think about it later. So then we moved from this little place to a building that Obaid had already started planning in the Agriculture University, and he had recruited a private architect, Raj Rewal, from Delhi to build a government institution. And it was only Obaid who could have pulled something off like that, because Raj Rewal had through his tantrums, Obaid held firm. The government, of course, refused to pay. So, there was all kinds of, drama, but Obaid got this building built in a matter of 4 years, and had able people to assist him in the dean and the director, Vijay, dean and the head of academics. And we soon built a thriving academic institution in this little corner of the Agriculture University. It's a beautiful part of the Agriculture University. It Today it's surrounded by all kinds of buildings and skyscrapers and things like that, but at that time there was nothing. It was farmland. And what you see at the back here is in fact pristine forest that was part of the Agricultural University and still is. But in the front of this side, it's now an urban jungle. And then, many of us got involved in engaging with running this institution. And very soon we had an institution where we were practicing biology across scales, from atoms to ecosystems. We were looking at various aspects of biology. And this was taking place with the intersection of biology, chemistry, and physics. People studied mechanisms that led to cancer, mechanisms of brain function. Neuroscience was extremely embedded because to study the brain, you need to study the cells and its structure and its organization and its complexity. So it was really highly interactive and interdisciplinary. So studying the brain was, also very, very, well embedded in this place. And there was an amazing sense of also the incredible space that we had begun to engage with and live in. So we had little more than 4 rooms. We had like 40 rooms now, and 40 labs could be accommodated in this place. And we were also building new structures. The atmosphere was incredibly collaborative. We had an engagement I'm just drawing lines that show the various collaborations that were present on the campus. But this wasn't just related to things on the campus. It was also in doing science across the country with various institutions as well. So here's, in fact, one of my colleagues who works on population genetics of tigers. And she discovers these famous albino tigers. And also, using traits from these tigers, she discovers how they move between different tiger sanctuaries. So some of the work that happened at NCBS also informed how corridors for animal movement need to be set up, because by following the genetics of tigers, you can follow where they came from. Just like by following the genetics of, looking at the genetics of extant humans, you can find out where they came from. So using the same tools, Uma had identified how these tigers can move, and that informed a lot of wildlife policy as well, or at least policy for building various roads which cut off different sorts of ecosystems from each other. Right. So science was also happening with huge collaborations across the globe as well. NCBS was beginning to be recognized as a small but powerful place for research. And in 2009, it was engaged in doing science, which was amplifying its links with a variety of institutions in India, nationally, globally. And there was also a connect to society that was building, but we were very small. We were, at this stage, about 20 faculty. So 20 years plus of research at NCBS had led to some sense of a unique flavor of doing science amongst many of our colleagues. And Obaid's mantra was that you would only pick the best. Who, well, of course, it was the best who also wanted to come and work in Bangalore, in a place. So we had a very eclectic group of people who had a variety of reasons why they wanted to be working in Bangalore, but who were also extremely good. And at this time, 2009, there was a growing sense that science needs to make a contribution, well, at least science that we did, which is TIFR, Tata Institute for Fundamental Research Supported Science, needs to make a contribution to the general, public good, many of us said, well, science itself is for public good. So why do we need to make any more of a contribution? But of course, that doesn't satisfy politicians. And then at this point, because we had groups of people working across various aspects of developmental biology, Dr. Ban, who was the secretary of the Department of Biotechnology at that, he had just become the secretary of the Department of Biotechnology, was shaking things up. And he said, and he became very keen of embedding deep science in medical institutions. And he was finding it difficult to make the connect of taking biologists and pushing them into medical institutions. So what he thought he'd do, and at this time, 2009-10, the idea of stem cells was beginning to emerge. People were beginning to recognize that this could be the next wave of therapeutics. So Ban held a carrot. He said, look, if you, as developmental biologists, which is stem cells are nothing but something that a developmental biologist would want to study in a living system, but in a therapeutic context, it's the cell that's taken outside the system, and you look at the stem cells that emanate in a dish. Ban had decided that he would hold this carrot of a stem cell institute and ask NCBS to participate in building India's first stem cell institute. So in fact, that's what happened. He again got hold of Vijay, who was always anxious to build new things, and he told Vijay, look, I've set up the Center for Stem Cell Research at CMC Vellore. It's a group of medical people working on stem cells in Vellore at a medical hospital, but I want biologists to be working on stem cells. And Vijay promptly offered our ecosystem, our environment, and in some sense, the rest is history, because we built the first institute for stem cell biology or stem cell science and regenerative medicine in next door to our own building. And then what happened was we decided that if we were going to build something that had such close links to application, we also needed a way to drive these applications out and build some pull, if you will, of biotechnology applications. And we created an incubator of sorts. We created I think this was at that time perhaps India's first biotech incubator. It was called the Center for Cell and Molecular Platforms, where this place, the C Camp, was in fact the brainchild, if you will, of Ramaswamy, and he got myself and Vijay and Taslim to become on the board of directors of this organization called CCAMP, which I think is also, in some sense, produced another institute in our vicinity. So let me tell you a little bit about InStem and what it does. So InStem is something that we call the Institute for Stem Cell Science and Regenerative Medicine, and its organization of science is somewhat different from that of NCBS. I mean, InStem is built stone's throw away from NCBS, and in this again fabulous building. It works on a variety of medically relevant questions. So cardiovascular disease, brain development and disease, chemical tools to address disease. That's the more cell-fate related structure. And there's inflammation, if you will, and also a place that is a translational unit that is in the center of the Christian Medical College in Vellore. That's also part of this structure. So InStem, and each group at InStem, it consists of a number of individuals who work on related problems. So they work as a team and as a group. And in some ways in academia, that's not the norm. So again, we were trying to experiment with structures of doing science in multiple ways. So, the research was multi-institutional, and very soon the Tata Institute for Genetics and Society, or the Tata Trust, wanted to build a new institute in the country, and this hadn't happened in the Tatas for research anyway, or for academic purposes, for 30 years since they built the Tata Institute for Social Science. So they approached us and said, we are trying to build something new. Will you be a host for this place? And we said, well, let's try it out. And we created a center at INSTEM with the Tata Institute of Genetics and Society between us and the University of California in San Diego. And so we created a center to advance the use of genetics in addressing societal problems. So that was the Tata Institute for Genetics and Society, or TIGS as we abbreviated it. And it came to being on our campus. So that was INSTEM, and then the Center for Cellular and Molecular Platforms, which in some ways is also part of, as I said, the translational arm of our local ecosystem. Today, it provides access to extremely high-end technology resources to both academic and industrial research operations, or even people who want to just go and use these technologies. I mean, they're an extraordinary suite of technologies that I think would be one would be hard-pressed to find anywhere in the world in one location. And these resources were, of course, because there were two life science research institutes side by side, with a lot of resources that have been put in through public funds, and we wanted to make them available in a large scale, and CCAMP was doing this. And very quickly, this is the number of startups that began to arrive and incubate in the laboratories that were set up at CCAMP. So today's CCAMP has about 410 startups that it has itself funded and incubated and accelerated and advanced. About 90 life science products that have gone into the market with shares, with monies on this order. And it, of course, has helped over 3,000 various startups in a variety of different ways. So in some ways, we had created an engine of taking science out from the campus by inviting startups to come and work in our neighborhood. In fact, I must confess, it's been hard to get rid of some of these startups because they just want to continue. But we have to find ways to send them off. So this resulted in the Bangalore Life Science Cluster, which I think today is a fairly sustainable ecosystem for the life sciences, if it is going to be if the basic stuff is going to be sufficiently funded. The startups, of course, all of them struggle because they get funding which is highly answerable to their investors, but basic science is what fuels them. In fact, many of our young scientists who work on our campus or join many of these startups or create some of them and go off and build value which is economic value. But the science that is being done on our campus also requires funding, and I think in some ways the sustainability of that ecosystem is what I think we'll have to think about going forward. Today there are about 1,000 scientists who work at this interface on our campus, and there's a new set of leaders now. There's Shashidhara, who runs NCBS since I stepped down. There's Manisha, who's taken over the running of the Institute for Stem Cell and Regenerative Medicine. And there's Taslim, who was the CEO at that time, and now he's become the director and CEO at the Center for Molecular platforms and is an extremely dynamic character. In fact, he also happens to be from Baroda. So that's really quite amazing. But we actually met in San Francisco, but he happens to be from Baroda. And then there's Rakesh Mishra, who was the former director of the Center for Cellular Molecular for CCMB, whatever those, in Hyderabad, who now is the head at TIGS. And together they make a fabulous team, and I think there's a fabulous future going ahead. I think, and I will underline, as long as we have a sustainable ecosystem to fund the basic sciences. So the campus looks, quite resplendent. It's quite beautiful. And in fact, we had a visitor recently, and she said that other than the SOC, perhaps the most beautiful design and research institute here is at NCBS in Bangalore, dedicated to understanding basic science in India. And I must say, even though I participated in helping to build it, I must confess it is an incredibly beautiful place. But it's not just beautiful. I think it's because we are an ecosystem of variety of different structures that work at different scales of the individual laboratory, the teams, and also startups and entrepreneurship. We can take on very large programs and build out activities that can have impact that would be way beyond the capability of a single research structure. And this is where I think the life science cluster or the ecosystem of doing science in this sort of distributed way becomes very exciting. So I won't list. I just listed out some of these activities that we're doing, but I'll take you through some of them. So one of them is in fact what we just managed to build out as a Center for Brain and Mind, very generously supported by the Rohini Nilekani Philanthropies, to study what I think is perhaps going to be an incredibly difficult thing in biology, but it needs legs and it needs support for, I would say it's maybe tens of years, if not more. And that is to study disorders of the brain, disorders of the mind. And that's where our partnership of the NCBS with the National Institute of Mental Health and Neuroscience, over 20 years of collaborations, resulted in building us a cohort of psychiatric patients that are deeply affected by 5 different clinical disorders, that of schizophrenia, bipolar, addiction, dementia, and OCD. So there are 5 different clinics where deeply affected patients and their families, who also have multiply affected people, have contributed to be part of a cohort, which is a unique cohort because of the unique population structure of our country. So we have, if you have familial diseases of which have complex genetic origins, having cohorts of this kind provides a resource that is just not available anywhere else in the world. So this Center for Brain and Mind came about because we were able to construct these, larger than individual institutional structures to create what I think is going to be the most exciting discovery biology in brain disorders. And because we have the patients, they have contributed their cells, so we can make stem cells and actually understand ways by which one might be able to modify or modulate these crippling diseases. So this is something that we actually, as a life science cluster, we're very excited by. We also study chemical ecology of our landscape in the Himalayas, in the Western Ghats, and try and understand the interactions of plants and animals and humans in terms of the chemistry, or the chemicals that are being connected between them. And what has been at the core of many of these things is our ability to reach across and bring the theoretical sciences, whether it's physics or computer science or even even game theory, which is another aspect between mathematics and computer science, into the midst of doing all this biology. So embedded in the ecosystem of life sciences, we have a number of physicists and theorists who are sitting inside this space, being able to unpack very complex phenomena using various theoretical perspectives which have hugely informed the kind of biology we do. And I think most exciting role, in fact, for young people has been a master's in wildlife biology and conservation that we have now had for the past 20 years, and try to create a cadre of individuals who would look at the wildlife and ecosystems of our country and create informed ways by which one may be able to engage in conservation. So, so this program, I think, is again would have only been possible because we had created the structure to support various engagements. We're also building a sustainability forum where we bring a number of different organizations working across sustainability matters in the Bangalore context, and have created a forum where we can talk across each other and in fact come up with ways by which we connect different organizations to work on sustainability issues, which I think is the most vital thing we have to do right now. Well, I think I'm going to say there are many community building efforts that have been possible. And beyond research, the campus now is able to construct ways of engaging with society that I think needed a home. So when Obaid passed away of a very sad and tragic accident, he was recovering from an illness, and he had been hit by a young scooterist, and he fell down, and being a very tall person, he cracked his head on the pavement and didn't recover. So we lost, I think, an amazing person. But when he passed away, this was his office, by the way, we felt it would be nice for us to create a place where we could bring the history of contemporary biology and science in India and create not a temple for Obaid or a shrine for him, but create something which would have brought an intellectual engagement with the process of doing science. And I think that's what has happened at the NCBS. The archives at NCBS is now a place where we have research papers of a number of amazing people who have worked in contemporary science in the country. Well, of course, we have Obaid's papers, but we also have papers of M.S. Swaminathan, we have papers of K.S. Krishnan, people of Sharat Chandra, we have and papers that are coming together, and Louis Coleman, who's also inspired the agricultural revolution here. So it's sort of been really an eye-opener to find people wanting to come and contribute and also engage in understanding how science was conducted in our shows. So that's the archives at NCBS, and it's a really exciting place. And I, of course, would like to say it'd be fabulous if many of you would take the time and opportunity when you visit Bangalore to pop in at our campus and see, or verify whether any of what I've spoken is fact or fantasy. Right, so I think, I'll say unabashedly that we have one of the best research clusters in the life science. It's perhaps, it's been an incredibly, amazing thing for me in particular to be part of this journey and helping to create it. But it's taken 30 years and a whole army of incredibly dedicated people who have believed that you could build something of this nature. And I think it takes very little to get rid of it. It takes, all you have to do is shut off the tap on doing fundamental science, and the rest of it will wither away in a matter of a decade. And I'm afraid that's precisely what's happening now. So I mean, in some ways, I think it will be very important for us to commit to building such a place and keeping it going, because it has taken a long time to build, and it'll take an equally long time to recreate again. And I think that's something we should be aware of. And if that idea doesn't convince you, when we had the need for such an institution at the time of COVID, our campus actually pivoted because we could. We had all the means at our beck and call. We instead of writing diyas and banging thalis, ended up actually going and doing stuff. We actually ended up going and doing work that was related to COVID. We actually set up the first testing services for the city, the state of Karnataka. We were, terribly engaged there. We actually tested more than 2 million samples in testing facilities that we had to build from scratch. We had to create the ecosystem for that in Karnataka. Genome sequencing of the virus. And what was more, and I think this was where we understood the power of the the startup and the small, medium-scale enterprises. CCAMP was engaging with many companies, as I mentioned, with startups. And we took on a mission to create an indigenous diagnostics, which had indigenous chemicals. Because initially, if you remember, the testing kits and the diagnostics for COVID were incredibly expensive. Very few could have it, and the government was spending huge amounts of money. So we built a platform which would hugely minimize the expense on this, and produced capacity of 1 million tests per day, which could be done at less than 100 rupees per test. And this is because we helped the companies indigenize all their reagents. So what I'm trying to say is that the capacity that we had on the campus allowed this possibility. And I think this was something that we were really tremendously grateful for having built such an infrastructure in these difficult times. Of course, it was very difficult to work. People were, getting infected with COVID. People were, some of our colleagues actually passed away, and it was really terribly tough time. But this was, I would say, the realization of the value of having built something like this. And this is something that can be replicated in many, many geographies in this country. So, okay, but I'm a scientist. I was just part of helping to build this ecosystem. So I'm going to end my lecture by coming back to my true love, and that's the membrane of a cell. So I'm just going to finish by talking to you about this creature that we saw in the beginning, and the motivation for me to actually digress and tell you a story about where the work is being done. And what I want to do is just tell you what we have found. I mean, in very brief and hopefully, shouldn't say simple, but hopefully understandable terms. Okay, so how am I doing on time? Yeah. So I'll be a few minutes. Yeah. So what we ended up doing is trying to understand, as I said, how does the cell sense. So we need to understand how the cell senses its outside, and for this, the cell, has its own inbuilt sensory system. It has molecules. I can't point anymore. Anyway, I'll use my pointer here. So it has these sorts of molecules which are proteins that are embedded in this sort of almost unrecognizable, boring little lines. So these proteins, which are called integrins, they sense the physical and chemical world for the cell. And if you can sense the physical and chemical world of the cell, the texture of the cell, the environment outside, the chemistry of the surfaces, or smell the molecules that have been, say, excreted by the bacteria, the cell will then have a sensory system to look, at least at that level of chemistry, of where it needs to go, and respond to it. And it does respond to it by creating this massive machinery when it engages with the outside world, this massive machinery at the inside of the cell. So if you remember the cell crawling about, there's a molecule that's sitting outside, sensing material coming from the outside, and then it produces this incredible response at the inside of the cell as soon as it is engaged with its sort of pieces of information. But it turns out that the cell has many such receptors, and they just make the cell distinguish between all these different sensory modalities of the chemistry, of the substrate, of its stiffness, and also of time scales. So the cell is computing all this information at the membrane. And what we discovered is that that these receptors are sitting embedded in this fluid membrane matrix. Now, if any of you have thought about a fluid system and you have something present, a perturbation present locally, that perturbation will dissipate very quickly in space and time. And effectively that fluid can carry no memory of that perturbation, right? Although, of course, there are all kinds of people who tell you that water has memory, but when you make an observation, you don't find that memory because it's a fluid. I mean, that's the property of a fluid. And what we discovered is that the cell, the membrane of a cell, in fact, completely belies this fact that it is supposed to be a fluid. In fact, it doesn't even respect those basic rules of physics. And it literally constructs a system which, for example, when information comes from the outside, it produces this whole kind of machinery inside. And then it feeds that information into the membrane, pretending as if it were not a fluid matrix. And what it does is that it constructs what we call our energy-driven structures, which carry information of what it has sensed. And it constructs those pieces of information into organizations that it builds within this fluid and captures it in terms of its structure and organization. And this means that this fluid membrane bilayer is going to be exquisitely patterned by information that is coming from the outside. And that patterning of information leads to a decoding of what the cell is sensing from the outside, and allows the cell then to make decisions about what it should be doing, because it carries a memory of what it has just received. And that memory is then erased, and it then reinforces it or not, and that decides what it can do. And then it's simply little programming rules that will allow the cell to go ahead and do something. So these are the sorts of things that we have uncovered, which are incredibly exciting, and are going to propel some of my future work. So I think if I can end, I'll say that understanding the cell membrane has required not only all the building of all these institutions and the correct environment to do science, but it also required the science itself. Physical-chemical studies on artificial constructs, insights from theory, and reality checks with experiments that we can do on membranes of living cells, and a full-fledged appreciation of how this came about in terms of the evolution of these systems. And that's another story, and it's just also very exciting. So I just want to leave you with this thought that the edge of a cell is a living, breathing skin. It's not a simple fluid collection of lipids and proteins, but it's a living, breathing skin. And it's exquisitely regulated by the machineries that it has inside, which are energy consuming. So, what one sees is these sensory systems orchestrating the structure of the membrane and encoding information that the cell uses for a variety of things. And of course, if one thinks about this can now begin to lead to a whole range of possibilities that could potentially have application. But the questions that we started with were not about application. They were about, curiosity. They were about how does a cell go and catch its prey. So I'm, going to start a new journey now, beginning to unpack some of the finer details of what we found. But I want to just say that I think, doing science has just been the most absolutely exciting thing that I've done. And being able to uncover something that cells do has been an absolute treat. And working with people who have made this possible is again, a real privilege, because I've just worked with the most exciting young and older people. I've worked with theorists, like dear friend and colleague of mine, Madan Rao, Ram Vishwakarma. So this is my current and ever-changing lab. I mean, they've just been amazing people to work with. And it has taken a lot of people to engage with us, have faith in us. And many of the people on this list here, you may know, have contributed deeply and fundamentally to our ecosystem, and more so in the recent times when funding for science has been drying up. So I really hope that we will be able to continue doing fundamental research, because that's what fuels the entire ecosystem of the startups and the economy that we hope will get going. So thank you very much for your attention. Wait for Shubha Mudgal. In fact, that's what I'm waiting for.
Satyajit, I think science without the time and the organizational structure, it cannot, deliver the results that we've seen. As Dr. Mayer said, during the time of COVID, whether it was a diagnostic tool or whether it was the actual vaccine, all of that came from the foundation of decades of science.